Thank you all for inviting me to talk this morning. I do have to say that in the last couple of days, I've come down with a really bad sinus infection, so it's entirely possible my head's just going to explode sometime in the next hour. Um, but one of the things I want to talk about today is the stress response and different ways that we can measure how birds are adapting to their environment or dealing with a stressor. So we're constantly having to deal with a changing environment. And I don't have to tell you about homeostasis. This is how our bodies maintain a constant internal environment. So we are fairly incapable of dealing with large changes in our physiologic states. A great example is pH. Our body's pH, blood pH needs to be around 7.4. If we get up to around 7.55, you're dead. If you drop below 7.2, you're dead. So we need to maintain this very narrow range of physiological environments. When our environment changes, external environment changes, there are different ways that we can adapt to those changes to maintain that narrow uh, area of life. And one type of change is referred to as allostasis. An excellent example of an allostatic change would be the stress response. So what is typically referred to as the hypothalamic pituitary adrenal axis in mammals um, is a great example of a genetically predetermined response to a stressor. Another type of way that we maintain homeostasis that is generally forgotten about is called rheostasis. Okay, so a rheostasis or a rheostat is you can think of it like the control of your room temperature. With the God, I just lost the word. Sorry. It's just a way of maintaining that narrow range of temperatures within our body. So a great example is how we deal with changing environmental temperatures. So one of my students, former students, Esther Oluwabinga, was from Nigeria, which is much hotter here. So even in the middle of the summer when it was 85, 90 degrees here in Indiana, she was cold and putting on sweaters. And but over time, she learned to adapt to our temperature. So her physiological set point was changed. And we do this too with physiology. So we can change our physiological set point, our physiological zero within that narrow range of survivable uh, states. And so, again, going back to the stress response, I want to talk about the HPA, so the hypothalamic pituitary adrenal axis. And that is referred to in mammals because there is a control over the stress response is in the hypothalamus. But in birds, it's different. And Wayne Kenzel has been at Arkansas has been pointing this out for years that there is a different organization, a different anatomy to the stress response in birds. So in birds, there is also the paraventricular nucleus of the hypothalamus, but there's also another area in the septum of the brain called the nucleus of the hippocampal commissure. And these release neurohormones on the anterior pituitary, CRH primarily from corticotropin releasing hormone, primarily from the nucleus of the hippocampal commissure, and another one, arginine-vasotocin from the paraventricular nucleus. These both act on the anterior pituitary to release ACTH, or adrenocorticotropic hormone, on the adrenal glands, which then release glucocorticoids. Okay, so when we're talking about this, the anatomy of the stress response, referring it to as the HPA is not entirely accurate. we really should refer to it as the septohypothalamic pituitary adrenal axis, or the SHPA, which is what I will do. And if you look at this nucleus of the hippocampal comma short, and these are photomicrographs from Wayne Kenzel's studies, you can see these cell bodies throughout the SHPA that contain CRH. So again, majority of CRH in birds is located outside of the hypothalamus. And if we co-stain for CRH and AVT um, in these brain areas, you can see here again. This is from Wayne Kenzel's studies. These cell bodies from our CRH, and the green fibers here are AVT. So the AVT neurons from the paraventricular nucleus in the hypothalamus not only project to and control a pituitary gland, but they also wrap around the CRH neurons. And if we'd had a similar photomicrograph from the PVN, you can see that the AVT neurons also are surrounded by the CRH axons. So there's a tremendous amount of crosstalk between these two nuclei in the septal region and the hypothalamus. And so how do CRH and AVT differentially regulate the stress response? We don't entirely know. There are studies from Wayne's lab of ferromedicine reported, and some studies out of my lab that suggest that the CRH in the NHPC is more related to a chronic control over stress, whereas the PVN is more related to an acute response, where the NHPC is more maintains and related to the chronic response. But we don't really know. That we need a lot of more studies on that. Um, but going back to glucocorticoids, one of the things that is generally forgotten in animal science or not addressed is that glucocorticoids generally are not about a stress response, right? They exist daily. They're constantly working in our bodies. Um, and their primary role is in the control of metabolism, glucose metabolism. And so it is possible to see changes in corticosterone or cortisol in mammals without there being an actual stressor. And a great example of this is something I did many years ago. I was doing a study on ducks. We'll just say it was somewhere in Europe, and completely unrelated to stress, but there was a stressor in the environment. It was a huge winter storm. Ducks are raised in curtain-sided barns, and these curtains were slamming up against the side of the barn. There was thunder, lightning, and heavy winds for days, and rain. And we were measuring blood and over time, and just had these huge levels of glucocorticoids, corticosterone in these two barns. And the study was on light cycles, which is not terribly relevant right now. But we went to another barn that was not experiencing, further away, that was not experiencing these conditions. And this barn, right, so you can see these birds, they were just horribly pecking at each other, which is kind of relevant since this is a PEC symposium, I guess, to show that this horrible amounts of feather pecking. And ducks are very unlike chickens. Chickens go after each other's heads. Ducks go after wings and tails. Very, very important species difference in feather pecking. Um, excuse me. And but we went to another barn that was not experienced, and all outward physical condition and behavioral observations was a perfectly happy barn of ducks, and measured their court at the same times. And this graph, it makes it looks like it's zero here, but it's not. It's just because of the y-axis, but very low levels. And these were all grow out or meat ducts. And just because this is the way my brain works, I wanted to go to a different type of duck barn. These are developers. And so developer ducks are young ducks that are um, being grown to become breeders. And so it's the exact same genetics, exact same strain. The only difference is how they're raised and measured them at the exact, and again, this by all outward appearances, a very happy, wonderful group of birds. And their baseline was different, significantly higher than the excuse me, than the meat ducks, but again, very low levels. So now, if we look at this purple line compared to the green, if that was the only thing we were looking at, a lot of people would say, "Oh my gosh, these developers are stressed." No, they're not. By all other measures, behavioral, physiological, these were very happy birds. Excuse me. So the question is, why are they different? Why is this higher than the grow out? And the answer is quite simple. The big difference between them is how they're raised. The developers are, excuse me, the developers are feed restricted, and this was during a feed restricted time. That doesn't mean they're stressed. That means in order to maintain glucose homeostasis, the glucocorticoids, corticosterone levels are raised. So right here, the difference between the green and the purple bars or lines are an excellent example of a rheostatic change. This is a change in the physiological set point of corticosterone. Whereas the blue and red lines, this is an excellent example of an allostatic change, right? So this is a stress response. So it's really important, when we're looking at corticosterone that we not overinterpret. And this is done quite frequently in poultry science, unfortunately. Another very important aspect here is, if all we did was look at one time point, we can be misled. So when we're looking at blood values of these hormones, it's really important that we look at multiple time points. All right, so what else can the brain tell us? And so Anna just gave an excellent seminar talking about neurotransmitters, which is what I'm going to talk about for the next few minutes as well in birds. Um, excuse me. So we talk a lot in animal science about affective states and what are some ways of measuring them. And Anna's already given us an excellent um, talk about dopamine and serotonin. So I won't go into a lot of that, but she had already talked about serotonin. It's involved with mood regulation, sleep, memory, as well as social behaviors and other peripheral effects. And serotonin, I want to point out that the vast majority of serotonin is produced here in what we call the mesencephalon. This is the midbrain. And this is a picture of a human brain because I can't find a similar one on bird brains, and I'm too lazy to make one of my own, or too artistically incompetent, actually. And but serotonin has this little serotonin fingers involved with all aspects of behavior, emotions, and homeostatic controls. So it is a very complex system. Dopamine is also very important in emotions, reward, motivation, but also in aggression. Very, very important neurotransmitter associated with aggressive behaviors and also movement, as or ambulation, as Anna already described. And most of the dopamine is also made in this midbrain, the mesencephalon. There are some others in the hypothalamus and a few other areas. In birds, this is a little bit different. This area, the ventral tegmental area, is actually divided between sort of the rostral and a caudal portion of the mesencephalon, so just a slightly different anatomy. But overall, the serotonin and dopamine systems in the brain are very highly conserved across phylogeny. Okay, so we can talk about these functions, not just with mammals, but with birds, fish, reptiles, very, very highly conserved systems in terms of their control over emotions or mood, or if you will, affective states. So I want to tell you a little bit more about how we measure it in my lab. Um, Anna already gave us a nice description of the synthetic pathway. So, serotonin is made from the amino acid tryptophan. The rate limiting step of um, serotonin synthesis is this enzyme tryptophan hydroxylase, which then with decarboxylase will make serotonin. Serotonin can then be broken down by different mechanisms into its primary metabolite 5-HIA, 5-hydroxyindoleacetic acid, which doesn't matter. But when you look at a synapse, serotonin can be involved in seen in different things. So this tryptophan is packaged up with the tryptophan hydroxylase up in the cell body, and then it's transported and synthesized as serotonin as it's being moved down the synapse or down the axon, and then it's stored down in the synaptic bouton in these vesicles, usually. And so serotonin in any given synapse can either be in these vesicles, it can be released where it acts released into the synapse where it acts on its receptors. And serotonin has a boatload of different receptors. So it's very, very complicated. But it can also be recycled by these reuptake transporters into and reused, repackaged to be reused later, or it can be broken down into 5-HIA in the presynaptic membrane. It can also be broken down by the postsynaptic membrane into 5-HIA. So if you think about it, if we just cut out a chunk of this tissue and measure serotonin, we don't really know what that serotonin is doing, right? So serotonin itself, it can be stored, it can be in the synapse, it can be broken down, it can be active, or it can be recycled. There are also these glial cells that surround these synapses, and they also will take up serotonin and do, from a scientist perspective, a lot of really annoying things. So when we want to think about the activity of serotonin, in my lab, we like to use this ratio of the uh, metabolite to the neurotransmitter. Okay, and it's what we refer to as the turnover. And this ratio is a indicator of the synaptic activity of the neurotransmitter of serotonin. Okay. And so in general, and this is not my crazy idea, this has been around for many decades. In fact, the first time I started reporting on this was in Wayne Kenzel's lab back in the 1990s. And yes, Dr. Weimer, I'm old, I understand. But if we look at an increase in serotonin activity, or excuse me, serotonin turnover, that is generally related to a lower activity levels of serotonin and is generally reported in many, many species over many different phyla as a reduction in affective state. Okay. Whereas a reduction of a lower levels of serotonin turnover is related to higher serotonin activity and a generally improved affective state. And the best way to think about this is with human medicine and veterinary medicine, right? So there's a $1 trillion pharmacologic, pharmacy pharmacological industry related to increasing serotonin activity in our brain, human brains, and our pets. Okay. And these are all the SSRIs, the serotonin selective reuptake inhibitors. So these drugs act to reduce the metabolism or catabolism of serotonin, stop it from being broken down and increase its synaptic activity. So when people are depressed or they have poor sense of well-being, they can go on these drugs and that increases their serotonin activity, which improves their sense of well-being. Okay. And so we can take advantage of this in animals by measuring serotonin activity to get an idea of their relative affective states. And one of the things I want to point out with this trillion-dollar pharmaceutical industry is there are, you know, dozens or a dozen, 20 different drugs that do an excellent job of altering serotonin activity in the brain. Not a single one of them is here, take a pill of tryptophan. It doesn't work that way. All right. So what's interesting is when we talk about very young animals, this idea of increase or decrease serotonin activity is somewhat reversed. So in really young animals, it can be the opposite of what I just described. There's only a couple of studies that have reported this. So the jury is still out on this. And personally, I can't decide if I think this is a really, really cool effect or really, really annoying. But there is that possibility that serotonin's activity on well-being, that individual's well-being may be reversed in very young animals. So dopamine has a very similar situation. It is synthesized from the amino acid tyrosine by tyrosine hydroxylase. Again, that's the rate-limiting enzyme, rate-limiting step. Dopamine can be, excuse me, broken down into three different metabolites, as Anna already explained, the HVA, 3-MT, and DOPAC. And again, we can look at dopamine. We can get an indicator of dopamine activity by taking a ratio of the metabolites to dopamine itself. And essentially, dopamine, again, remember, it's associated with aggressive behaviors as well. So in animals, an increase in turnover refers to it's being metabolized more. There's a decrease in dopamine activity. So that can be related to a more positive affective state, and the opposite with a decrease in turnover. And again, with birds, or excuse me, with very young animals, the situation can be reversed. All right, so how do we put all of this into practical use? And so I want to tell you about a study that was just published by one of my graduate students, Isabella Van Sigern, and where she was looking at how an isolation test would affect these neurotransmitters. Okay, so she wanted to address the question as whether or not how age would have an impact on this isolation fear response and due to changes in these neurotransmitters. So she looked at birds at 12 days of age, 12 weeks of age, which are, and these are all in layer hens. And at 12 weeks, these are pullets, so they are sexually immature. She also looked at mature birds during peak lay, 26 weeks of age, and then geriatric birds. So these were birds that were coming out of lay at our farm here at Purdue. And she put them in this isolation chamber. And another part of the study that I don't have time to go into is she was also analyzing vocalizations that occurred. And so in order to analyze vocalizations, are put in this chamber with anechoic foam. So this this is so we can get pure sound, no reverberations, no echoes and such. And control birds were just pulled out of their home cages. These were in conventional cages, pulled out, or excuse me, not conventional cages. These are in enriched cages, and they were immediately pulled out and euthanized and assayed. She also compared birds in lit isolation chambers and as well as dark. And that happened because she was a brand new graduate student in my lab when she did this study, and I neglected to tell her to turn the lights on. So it turned out her first group were done completely in the dark, and we're like, what the hell? Let's roll with it. So we did light and dark conditions throughout the rest of the study. And we collect the brains and micro dissect them down further into rostral and caudal mesencephaly. And if you remember, that's the uh, source of all of the most of the serotonin and most of the dopamine in the brain. And we dissect it down into rostral and caudal components because there are different aspects there. The diencephalon. Now, I call it the diencephalon here, but in reality, we also include that nucleus of the hippocampal commissure, so it is a part of the septum as well, just to get that SHPA response or level or areas. Um, but we also dissect down into the telencephalon into medial and lateral components. The medial component is primarily the hippocampus, which is most well known for its involvement in memory formation. But as the previous talk described, it is a very important component of the limbic system. And the lateral telencephalon includes areas in the bird brain comparable to the nucleus accumbens, which is associated with motivation, reward, addiction, um, and also the amygdala. The amygdala in birds is quite different than it is in mammals. This is the area that's formerly known as nucleus tenei in birds, now referred to as amygdala, but it is just one component of the amygdala. It's not the entire amygdaloid complex as Anna referred to in the last talk. And of course, we did statistics, right? Lots of statistics. So if we're looking again at just the turnovers, we found this wonderful age-related difference in different areas where we saw increases in serotonin turnover, particularly in the young birds, in the chicks. And again, this is probably their when they were isolated as young birds, we're thinking that this is actually improving affective state because maybe this is defense against predators. Yeah, we don't know. Um, in pullets, we didn't see any changes at all in serotonin turnover. And this was very interesting, and I'll tell you a little bit more about that why. But apparently, the tweens brain, the tween brain just doesn't relate to anything. Um, but at peak lay and older birds, the serotonin activity is definitely telling us that when in isolation, they have a relatively poor affective state or poor moods compared to controls, which makes sense. And all of the vocalization, I don't have time to go into, but supported all of these findings. And what's really important here is that the birds are all expressing fear, I guess, but they're expressing it in different ways with different ages. When we looked at dopamine, we really didn't see any effects on anything except during peak lay, would suggest that they were happy, but they're not. Other things are telling us that they're not. So this is most likely not really suggesting that they have a better affective state, but this is more probably assist a matter that related to their motivation. So the serotonin is telling us, yeah, they have less affective state, but they're probably less motivated to do anything about it. When we looked at court in these birds, we found very interesting. So again, at peak lay, very obviously that they're showing a stress response with court, but we didn't see it at any other ages. And so the big importance here is with gonadal hormones. And so the lack of stress response in the younger birds has been shown many, many times by many different labs, including Darren Karcher's lab here at Purdue, with a study done by Megan Abraham in pullets. And so this is very, very interesting that the birds, the court response, if all we did was look at court, we'd say, yeah, they're not stressed at all. But the brain is telling us otherwise. So just like in humans, the brain serotonin activity can reflect the relative affective states. Um, when you're looking at control birds, but court is probably not quite as reliable as an indicator. Okay, so all of this was about an acute stressor. What about something a little bit more long term? And now let's go into something that's a little bit more relevant to industry, um, and that's heat stress. Okay, and now I'm also going to switch to ducks, a study on ducks. So ducks are raised in these curtain-sided barns, predominantly around the world, and so whatever temperature and humidity is outside is generally what's happening inside. Here in the United States, ducks are about 85% of duck production is done on Amish farms, and Amish uh, don't typically use electricity, so ventilation, tunnel ventilation is out. So it's all through natural ventilation through these curtains. And whenever heat gets up above 35 degrees Celsius or 95 degrees Fahrenheit during the day, and importantly, does not drop below 85 degrees Fahrenheit or 30 Celsius at night, this, for a long period of time, this really has uh, bad effects on the ducks. So ducks are very heavily feathered. Their feathers are a very important part of the industry. They have that very thick layer of down, which is great. That really does help protect them with heat stress until we get conditions, long-term conditions like this, and then it works against them. And I don't have to show you all of the data about how when we cause these conditions over a 3-week period, how it decreased hatchability, fertility, welfare rate. This, we've published this a few times, and it's kind of a silly because farmers have known this for 100 years that this is really, really bad. So I want to focus on some other aspects. So when we look at court over many different time points during a 3-week stress, and then we also gave a 2-week period of recovery afterwards, we get a very typical court response. First off, we see a very important sex difference between the drakes, males, and the hens. Um, but we get a wonderful peak in the court response early on in both sexes, um, that then wanes and comes back down to baseline. And this is a phenomenon that is absolutely well established for since the 1980s, with chronic stress. Okay, and so if all we did was collect one time point here or out here at the end, we'd miss it. And we're not seeing quite as big a response here, and in the drakes, again, this we may have missed it. We may have been more around here relatively, but we are seeing the increase at a different time point. And again, we've shown this many, many times um, in these studies. And so now the question is, okay, so court is the body's response to stress. Okay, court does not cause stress. Court is the body's response. This is how we are resetting homeostasis or maintaining homeostasis under these stressful conditions. So does that mean that the birds are no longer coping out here when court is back at baseline, or is something else taking over? The the allosteric response. So Colin Skanes and I were talking about this a couple years ago, and he suggested that I start looking at another neurohormone called metenkephalin. Okay, so metenkephalin is one of the body's endogenous opiates in our brain. It is synthesized in part by the POMC gene. Okay, so POMC is most well known by most of you that one of its genetic transcripts is ACTH. ACTH is the anterior pituitary hormone that stimulates the adrenal glands to release cortisol. Okay, but alternate transcripts in the brain. So POMC itself in the brain is a neuromodulator, is a neuropeptide that has many different behavioral effects. But alternate transcripts of the POMC gene can produce all of these different things, including metenkephalon. Metenkephalon is also produced by not only POMC up here in the top panel, but by proenkephalin gene. And so all of these black boxes are different transcripts of these genes that can produce metenkephalin. And so one of the things that, I really want to point out that when we're talking about the brain, okay, so the previous talk and I have been talking about the biogenic amines, all of the dopamine, serotonin. We didn't talk about norepinepi, but amino acids are also neurotransmitters in the brain. Purines themselves, like ATP, right? We know what ATP is for energy, but ATP is also a type of neurotransmitter in the brain. There are neuropeptides. These are just four of them. There's somewhere on the order of um, well, let's just say billions and billions of them um, in the brain. Gases are also neurotransmitters in the brain, like nitrous oxide, carbon monoxide. These, we're familiar with their pharmacological effects, or death, but they also act as neurotransmitters in the brain. So the important take home here is that when we're talking about emotions and neuroscience and the brain, it's very, very complicated. And so we neuroscientists have to think about all of these. And so I guess the most important take home here is now you understand why we drink so much. Okay. But if we look at metenkephalin, let's go back and let me remind you of the court. So we got this early court response in both sexes, but then it wanes off and then drops back down to baseline at the end of this stressor and into recovery. So if we look at metenkephalon, it's having sort of the opposite effect. So it starts off at baseline, but then it raises, it increases when court is dropping back down to baseline. And this is happening in both sexes. And then after 2 weeks of recovery, everything comes back down to baseline. So there's a couple of possibilities here. Um, so Colin Skanes has recently reported that, during a stressor when court is high, if they inject metenkephalon, that brings it back down low. So metenkephalon could be acting as one of the agents that is bringing court back down to baseline. But metenkephalon could also be taking over its effect, the central effect to maintain homeostasis. Okay, so very, very interesting. Um, and something that we're looking into more. But again, this court, remember the court's being released high here. So what's happening? So these are the adult breeder ducks. What's happening with the eggs? What's going on? And what I'm going to tell you, I'm just going to summarize some work that was done by a former PhD student of mine, Esther Olowabinga, who's now working for research scientists in Neon Incorporated out in New York. And if you want to catch up on what she's doing with Neon, you can. She was highlighted in a story by the New York Times. So pretty cool. Good, great kudos for her. So one of the things we did early on with these heat stress is we were measuring glucocorticoids in the eggs, and we use mass spectrometry for all of our amino acid or our neurotransmitters and glucocorticoids, at least in the egg, and and we found some interesting things. So first off, we found that we did not, unlike was classically reported, we didn't find corticosterone in high levels anywhere in the egg. We found cortisol, right, which is typically thought of as the mammalian glucocorticoid, um, but we found it there, and we found it primarily in the albumin, not so much in the yolk. And so we're not the only ones that have reported this. Several other labs, including Caulfield and Padula, who, and what's interesting is that all of the labs that use mass spec to measure glucocorticoids in the eggs, we find cortisol. Those who use ELISAs find corticosterone. Now, what exactly is this difference? It's complicated, but finding it in the albumin versus yolk is important. And there was a study a while ago now by Rittenbacher, reported in Annals of the New York Academy of Sciences, where they injected radio-labeled corticosterone in chickens, and they actually found that it was accumulated in the albumin, not the yolk. So how exactly is that getting in there? I'll talk about later. But that's really important. But what is it doing? Does that court have an effect on the offspring? And so Esther looked at all the production uh, variables and didn't really find any effects. There was a slight reduction in the birds that from the parents that were heat stressed that had the higher court levels in the egg. Slight uh, reduction in birth, or excuse me, hatch weights, but that did not maintain throughout the production period. And we didn't find any effects on FCR, feed intake, body weight gain, anything. Um, but we did give an ACTH challenge to these birds, the F1s. And so again, we're not causing stress. We are simulating the response to stress, to a stressor, by injecting ACTH. ACTH being that anterior pit hormone that uh, um, stimulates the adrenal cortical cells to release cortisol. Okay. And excuse me, what we saw in the offspring of the heat stressed parents is that they had an exaggerated ACTH response, or court response to the ACTH, not only in the court response, but also in the heterophil to lymphocyte ratio, which is another indicator of acute stress, indirect. So that was very, very interesting. And we have actually shown this exaggerated ACTH response um, 3 times now in different studies related to heat stress. So very, very cool. Esther also ran a bunch of behavioral tests, and let's see if I can make this work. And if a picture is worth 1,000 words, and a video should be worth at least one grant. So these are offspring, the ducklings from our heat stress, and we like to use a laser pointer for as a novel object. Long story as to why that is, and typically don't really see it here, but the ducklings will actually chase it. We'll typically chase it and almost like a cat, so it's really cute. And so these were from ducklings from the control birds, and here's from the heat stress birds. And you can see very different behavioral responses. And, of course, once we graph it all out, there is the birds from the heat stress parents were significantly more fearful overall at every single age we looked at. All right, so that's interesting. So we have this increase in the hormonal stress response in offspring from heat stress parents, also an increase in the fear behaviors, and we did multiple different fear tests, not just the novel object, not just the laser pointer, and so that's interesting, and it's also coordinates very nicely with what has been reported in the wild bird literature. They also show increases in fear response and the court response in wild birds. And it is generally believed that this is a way for the parents to pass information off to their offspring, saying that you are living in a higher stressful environment and it's preparing them. So the increase in the court response and the HLR is preparing them for to maintain homeostasis in a stressful environment. The increase in the fear response in wild birds is believed to be a good thing because this is making the offspring take fewer risks, right? So they're more cautious, if you will, in this higher stress environment. So in the wild bird literature, these factors or these changes in the F1 are thought to be a good thing. Whether or not this is a good thing in our poultry remains to be seen. Um, it probably just depends on the management or what's going on in the barn at the time. Okay. So this is very interesting, but the increase in the court in the eggs and the change in the F1 phenotype is purely correlational. We neuroscientists, we really, really hate correlations, right? There's a bazillion different rows of dominoes and multiple parallel rows of dominoes that all fall between these things. So we wanted to test whether or not there is a direct connection between the increase in court and these responses. And I don't have time to go into all of these details, but this what we did was we injected glucocorticoids into the eggs and then did raise these as meat birds and looked at this. And this was a study done by a new graduate student of mine, Nina Wilson. And here's the bottom line is yes. And this is a study that's currently under review. Um, we, not we, Nina, perfectly by injecting into the eggs, a glucocorticoid into the eggs, she perfectly replicated that increase in the court response to ACTH. She perfectly replicated the HLR response to the ACTH that we observed many times now in the offspring of heat stress parents compared to controls. What we did not she did not replicate was the behavioral outcomes. So it's possible that this court is inferring some epigenetic effects on the hormonal stress response, but not behavior. So that's really, really cool. Okay. So I've been talking about cortisol, and I just want to wrap up by talking about that for just another minute. Um, Cortisol in birds is not new. Okay, this is not a new thing. Back when glucocorticoids were first discovered in the 1980s, everybody was focused on the fact that birds had both corticosterone and cortisol. And in fact, many labs thought that cortisol was a better indicator of stress in birds than is corticosterone. And so when I've talked about this lack of corticosterone effects in young birds, that's been shown in all kinds of different species of birds. In those birds, when they're not showing a corticosterone response, they do show a cortisol response. Okay, so very interesting. And even more recently, there have been studies in Karen Soma's lab up in Canada where he has shown that cortisol is produced not by the adrenal cortical cells, but in other tissues, but in immune tissues. And in response to corticosterone, cortisol is synthesized in these tissues and acts as a paracrine hormone to leads to the reduction in bursa size or thymus size in response to chronic stressors. So cortisol does exist in birds. It is an active hormone in birds in response to probably chronic stress and probably is doing a lot more things that we don't know about. These ideas, again, with the epigenetics of court, well established in wild birds. Um, And um, we are currently looking at this in ducks here as well. Okay, so we can't forget about you know the different aspects of the stress response. Okay, so there are multiple glucocorticoids, right? So we can't forget about cortisol. We can't forget that in birds, the anatomy of the stress response is different. Okay, it's not just an HPA, it is a septohypothalamic pituitary adrenal axis. Um, there are multiple glucocorticoids, including cortisol, that have a very important physiological role. And there's lots of other neurohormones, and the effects within the brains. Um, so, you know, we look at serotonin and dopamine, and these are very important indicators of an affective state, but metenkephalin may be important. Endocannabinoids. So, this is a whole nother seminar talking about the endogenous, THC, marijuana, neurotransmitters. Endocannabinoids, may be indicators of a positive affective state. And that's something I'm working on with Heather Neve here at Purdue. Mesotocin, which is um, the uh, the analogous or orthologous hormone, neurohormone to oxytocin in birds, or oxytocin in mammals. Okay, is mesotocin possibly another indicator of positive affective state? That's something else we're looking at. The potential epigenetic effects. So I'm working with Luis Brito here, looking at how that court in the egg is affecting multiple generations, the phenotypes of multiple generations. And what's really important is that these glucocorticoids, everybody measures them now. It's so easy to measure, which is unfortunate for certain reasons. But it's very uh, possible to misinterpret the effects. Okay, we have to remember the allostatic versus rheostatic changes. Okay, so again, we're looking at these possible epigenetic effects, and looking at serotonin and dopamine and how this has effects on aggressive behaviors in ducks, which is very different than chickens, and other unwanted behaviors. We're also starting to look at the gut-brain communication with Josh Light down at Arkansas, USD Arkansas. Um, and this gut-brain axis is something that's very interesting and very annoying about. There's a whole lot of arm waving about it. That's a whole nother story. And again, looking at these epigenetic effects. All right, so lots of thanks to lots of folks. I have a big, wonderful lab, lots of great people. USDA has been very generous to me over the last few years, and lots of great collaborators. And of course, the stakeholders who can't do any of this without them. And thank you so much.